you for being here. Today I will present an optical technique for the detection of bacteria in complex sample, and I will focus my presentation on Staphylococcus aureus detection in blood sample. So, bacteremia refer to the presence of viable bacteria in blood, which is normally a sterile site. It can be a very transient event, for example, after a small cut. But for more serious wounds or for immunocompromised people, the infection can worsen. Each year, 200,000 cases occur in the United States with a mortality between 20 and 50%. Among the causative pathogens of bacteremia, Staphylococcus aureus accounts for almost 20% of bacteria. So, as you have understood, bacteremia is a life-threatening infection which needs rapid medical care. But the physician, to give an appropriate antibiotic treatment, need to know the identity of the bacteria involved in the infection. To do this, a blood sample is collected. Usually between 5 to 10 milliliters of blood are required because bacterial load of blood is quite low. In general, less than 1 bacteria per milliliters of blood. Then this sample is diluted with an appropriated culture media and incubated to assess bacterial presence. If the hemoculture turns positive, a second step of isolation of the bacteria is necessary, and then a third step to identify the bacteria and to test its antimicrobial susceptibility. The whole identification process takes between 3 days to 5 days, and during this time the patient is receiving an antibiotic treatment which could be ineffective for the bacteria which is involved in the infection. So there is an imperative need to shorten diagnosis time. Our goal is to perform hemoculture and identification in the same time in the hemoculture vial in order to give reliable information about the bacteria involved in the infection at the end of the hemoculture phase. To do this, we propose to identify bacteria on an antibody microarray, and the biochip is constituted of a glass prism with a gold layer on its top. An antibody are grafted on the gold layer by an electrochemical reaction. Then a kind of reactor, which you can see here, is put on the prism, and biological recognition between bacteria and antibody will happen in this compartment. And the interaction between bacteria and antibody will be visualized by surface plasmon resonance imaging through this prism on an optical devices at the bottom of the prism. The detection of bacteria by SPRE is made by an approach named culture capture measure. In fact, the low number of bacteria initially present in the sample will start growing and interacting with the specific antibody. It will generate a signal, a localized signal on the spot of the antibody. On the image, we can see a bright zone on these antibodies. And you can see also on a graphic with a variation of reflectivity over time and an important response for specific antibody. So detection by SPRE is made in real time. It's a label-free detection and a multiplex detection. Moreover, it's a non-destructive approach, so it enables further testing on the sample after the end of the hemoculture phase. This approach has already been validated for detection of pathogen in food matrix, like milk or raw meat. And in blood sample, this is a proof of concept of the feasibility. Here we are able to detect 100 bacteria of Salmonella enteritidis in a few hours in diluted human blood sample. But we now have to see what happened with real pathogen involved in bacteremia, like Staphylococcus aureus. And what happens, and what we can see on this graph, is a problem of specificity with an augmentation of the signal on all antibody in the same time. This phenomenon is due to the presence of the staphylococcal proteina on the membrane of Staphylococcus aureus, and this protein has the ability to bind to the constant fragment of antibody. So, as you see on this scheme, you have some interaction are mediated by proteina and other by antigenic determinant. So it's difficult for us to say if an antibody actually recognizes its target or if interaction are only proteina related. To overcome this difficulty, we try to cleave the antibody in order to remove the constant fragment and to keep only the fragment which is able to bind to the antigen. What you can see in this picture in green is digested anti-Staphylococcus aureus antibody. They are successfully binding to their target with lower affinity than the whole antibody here, because the whole antibody has protein A and antigenic interaction. And when you do the same thing with a non-specific antibody, here in purple, there is no more interaction with Staphylococcus aureus, which means that the digestion protocol work and digested antibody is a solution to remove proteina interaction for the detection of this bacteria. Well, in conclusion, we can say that specific antibody are required for proper bacterial recognition, as a result shown. It influences both sensitivity and specificity of the assay, but with this condition, bacterial detection is feasible in a few hours, even in complex media like diluted blood sample. In perspective, we have to test alternative probes like chicken antibody, for which proteina have no affinity, or aptamer, for example, and after to compare all results with existing device for the diagnostic of bacteremia. I would like to thank my PhD supervisor, Johan Ropios and Thierry Livache, Diane Poulido for the work done together, the Professor Max Morin and Sandrine Boisset from Grenoble Hospital for sharing their knowledge about Staphylococcus aureus, and the CEA program Technologie pour la Santé for the funding of my PhD thesis. Thank you.
Thank you very much for this nice talk. What do you think will be the costs if such a will be?
For food pathogens, they already start to do the industrialization of the process, so trying to reduce the cost, maybe 50, I'm not sure about 50 euros per test or 40. Yeah.
But not cheap.
Not cheap. They are trying to reduce the cost, but the problem is the prism, which is costly, and the optical device a little also. But
the talk is open for questions.
Which antibiotic do you use to treat Staphylococcus aureus?
I'm not using antibiotic, but the problem for Staphylococcus aureus is the important resistance for antibiotic. Vancomycin is the last possible antibiotic. After, we are also trying to identify resistant determinants on Staphylococcus aureus, or to perform tests in order to give also information about resistance to physicians.
In fact, I asked you this question because I have a previous infection with Staphylococcus aureus.
Yeah.
Since 5 years, I work here in Germany, and I have this infection. They put me in the hospital separate, and they injected me with penicillin
Yeah.
for 11 days. And they completely
Okay.
Coming back to scale, is there also next
Yeah.
question?
Yeah. It's a very, very nice piece of work.
Thank you.
Very interesting. Is it fair to say that all pathogenic Staph aureus contain protein A?
Now, all all strain of Staphylococcus aureus exhibit different level of protein A. Maybe I have a strain which has a lot of protein A, but if I want to be able to detect all Staphylococcus, I have to do this kind of experiment. And maybe I will have the same problem with Streptococcus and protein J. So I have to think about it, yeah.
Next question.
Back to the scheme of gold. Why you
Use the microphone.
This one?
why
Which?
use gold here? What is the idea of using gold?
The gold layer is for the plasmonic, its property for the system of SPRE. In fact, the electron on the gold layer are well, 1 second. No. The electron on the gold layer are excited by the laser here, and they're producing a kind of wave on the surface.
Yes.
And this is why gold is an appropriate material to do.
Use it like for imaging.
Yeah, for
Measure
imaging.
light.
You can also use silver, but it's due to physical property of the gold that it's this metal that is used.
More questions? Please.
Yep.
Please use the microphone.
If you have studied the comparison between contamination of food, test of challenge,
Yeah,
and
yeah.
if the time is equal in
Yeah.
between food contamination and blood contamination.
Could you repeat? Sorry.
If you have
Yeah.
studied comparison between contamination of blood and contamination of food. Contamination of food, it's challenge,
Right.
challenge
Yeah,
test.
yeah. You have to compare with existing
If
data.
the time of both contamination is the same.
My device is.
Can you repeat the question, please? Because
I
no
think-
one has understood.
The time of development of the contamination of blood and contamination of food. Contamination of food, it's a challenge test.
Yeah.
Donc, it's the time of development of pathogenic species. It's
Same.
equal.
I think it's blood. It may be a little much longer because blood is not really good media for bacteria, and that's why we diluted blood in other. But the dilution of blood is not only for this precise detection. It's also does for hemoculture. When you have an hemoculture, blood is diluted in an hemoculture vial. In general, there is 1 volume of blood per 5 volume of media, 5 to 10. It's bacteria. It's really difficult for bacteria to grow in blood only because there is a lot of antibody and cells which interact with bacteria and try to eliminate it.
Because food,
Yeah, in food also, but food are also, for example, for raw meat, you diluted 25 grams of meat in 250 milliliters of media, I think. It's not exactly the same. Each application has its own protocol.
Yes, please. Yes, first of all, it's a very interesting subject. It is
Thank
very
you.
nice presentation too. What about the MRSA or massa in this point? Can this work plays any role in the MRSA and MASA? Because this is the one, I think, it's one of the main problems with the Staphylococcus aureus infection, right? So you didn't mention what can we do if this can help us in treatment or diagnosis of MRSA strains, methicillin resistance. I know it is, you didn't discuss it, but it's I'm just opening discussion, okay?
Okay. We are trying to differentiate between MRSA and MSSA strain. I did not present it here. By antibody, with only antibody, it seems to be difficult to have a differentiation between MSSA and MRSA. So we try to do with add of antibiotic in the media maybe and see if there is growth with antibiotic.
I understand from your experiments that you make this something like microchip,
Yep.
right, of mangold and bind antibody to this gold.
Yep.
And when this gold will catch, when this antibody will catch the staph organism, it will make some signals and you can catch it with your device.
Yeah.
It's a very good idea. But actually, to reduce the cost, that's one of my opinions. Because I know someone who makes research like this, but you know that when the gold particles increase in its size, then it changes its color. And you can see it with your eye without any devices.
Yeah. There is some publication.
Bind antibody to it and use it for detection also.
There is publication with Aptamer.
For example, site we use, for example, 10 nanometer and it's red color, and you can see it with your eyes, with naked eyes, without any devices. When these particles will group together, the size will be increased, so it will change to purple color, and you can measure it even colorimetrically if you want.
Yes.
So you can eyes to right?
I agree. I saw some publication with Aptamer on the nanorods like this. The one advantage of this technique is that it can be multiplexed. I can have 10 different antibody.
Yeah, that's good.
And maybe with the nanorods.
And it can be reusable?
No, not really. Not with antibody because it's quite fragile.
Yeah.
But with other probes, maybe we can reuse it.
Thank you.
Is there more questions? If this is not the case, thank you very much.